Hello, everyone. My name is Suganya Phantu, and today I will present our study entitled Predictions of Total Phenolic Content from Rice Bran Extract obtained by Ultrasound-Assisted Extraction using multiple mathematical models. The presentation is divided into 5 main parts. First, I will introduce the background and research problem. Second, I will explain the objective of the study. Third, I will describe the methodology, including the experimental design and model development. Fourth, I will present the result. And finally, I will conclude with the optimal extraction condition and key findings. The backright bran is a variable byproduct from rice processing. It contains many bioactive compounds, especially phenolic compounds, which are known for their antioxidant potential. However, the amount of total phenolic content extract from rice bran depends strongly on extraction conditions, such as extraction time, temperature, and the solid to solvent ratio. Ultrasound-assisted extraction is considered an effective and greener extraction technique because it can improve extraction efficiency while reducing time and energy use. Nevertheless, identifying the best extraction condition through trial and error can be time-consuming. Therefore, this study combines experimental design with mathematical and machine learning model to predict and optimize TPC extraction from backright bands. Next, there are 4 main problems addressed in this study. First, backright band contain variable phenolic compound, but its use as a functional ingredient is still limited. Second, the extraction efficiency of TPC is strongly affected by several process variables, especially extraction duration, extraction temperature, and solid loading ratio. Third, conventional trial and error optimization require a lot of time and money and may not be efficient enough to find the true optimal condition. First, the relationship between extraction variable and TPC may be nonlinear. Therefore, machine learning-based prediction is useful for capturing complex relationships and improve optimization accuracy. This study has 4 main objectives. The first object is to develop and compare regression and machine learning model for predicting TPC based on FCCD experimental data. The second object is to evaluate model performance using statistical accuracy measure. The third objective is to examine the residual behavior of developed model, especially in terms of normality and independence of error. The final objective is to determine the optimal extraction conditional that maximize TPC from backright band. In this study, the raw material was backright bran, which is a native Thai proteinous rice variety with a black pericarp. Pigment rice, including backright, is known to contain antioxidant compound, especially phenolics. These compounds provide health promoting property and makes backright bran a promising source of functional ingredient. The target response in this study was total phenolic content, or TPC. The main goal was to maximize phenolic yield while reducing extraction to energy and preserving bioactivity. The experimental design used in this study was the fake center central composite design of FCCC. Three action factors were investigated. The first factor was ultrasonic processing duration with level of 25, 13, and 35 minutes. The second factor was ultrasound extraction temperature with level of 13, 14, and 15 degrees Celsius. The third factor was its solid loading ratio with level 1 for 15, 120, and 125 grams per milliliter. The response variable was the total phenolic content obtained from each extraction condition. The machine learning methodology starts with the three input variables: extraction duration, extraction temperature, and solid loading ratio. These variables were used in the FCCD experimental design. After that, feature generation was performed to create additional features from the original variable. Modeling and feature selection was then conducted within each model. Three main models were compared: polynomial regression, attribute regression, and kernel ridge regression. For each model, the best feature set was selected using R-square, SSR-square, and IMSE. Finally, the best model was compared. Parameter optimization was performed, and the final optimized model was selected based on the best overall prediction performance. To evaluate model performance, three statistical measures were used. The first measure was R-squared, which indicates how well the model explains the variation in the observed data. The second measure was SSR-squared, which is useful when comparing models with different number of predictions because it accounts for model complexity. The third measure was RMSE, or loose mean square error. A lower RMSE indicates smaller prediction error and better prediction accuracy. Together, this measure helped identify the most accurate and reliable model. The residuals diagnostic analysis was also performed to check whether the model error behaved properly. Four aspects were considered. Normality means that the residue should follow a normal distribution with a p-value greater than 0.05. Independent means that the error should not be correlated. The means of residue should be close to zero. Finally, the variance of the residue should be low and stable. These diagnostics help confirm whether the prediction model is statistically reliable. This slide shows the feature selection result for the three models: polynomial regression, attribute regression, and kernel ridge regression. For each model, different number of generated features were evaluated. The select feature set was determined by balancing high predictive performance with model simplicity. The feature important result also show that interaction and polynomial term contribute to the prediction of TPC, indicating that the extraction process involves nonlinear relationship among the input variables. The results show that all three models achieved relatively good performance, but the best model was polynomial regression. The polynomial regression model achieved an R-squared value of 0.995 and SSR-squared value of 0.995. 983 and the lowest RMSE of 0.067. At the boost, ligation also performed well with an R-square value of 0.99, adjusted R-square of 0.985 and RMSE of 0.095. Kernel regression has lower performance compared with the also true model, with a square value of 0.962 and adjusted R square of 0.929 and RMSE of 0.181. Therefore, the polynomial model was selected as the final model for optimization. In conclusion, this study successfully developed and compared regression and machine learning model for predicting total phenolic content from backed-Lyban extract. The best performed model was the polynomial model, which provides the highest predictive accuracy and the lowest prediction error. Using this model, the optimal extraction condition for maximize TPC were identified as an extraction duration of 35 minutes and extraction temperature of 39.44 degrees Celsius and a solid to solvent ratio of 1:25. Under this condition, the predicted TPC was 18.77 milligram per diet. This finding showed that combining FCCD with predictive modeling and effective support the optimization of ultrasound-assisted extraction from background band. This slide presents the main reference used in this study, including work related to background bioactive compound, experiment design, and statistical model selection. The reference support the experiment design, model evaluation, and interpretation of the results. We would like to acknowledge the Faculty of Science, Energy and Environment, King Mongkut University of Technology, North Bangkok, Rayong campus, for supporting this study. Thank you very much.